Log on, tune in, find out. Another good idea from Cambridge.
I'd have a go at doing principal components analysis. And the problem is that PCA is really designed to work with vectors, and trees aren't vectors. So what am I going to do? Well, here's my solution. I'm going to regard principal components analysis as a kind of geometrical solution, procedure, and then I'm going to set up some geometry in tree space, do the geometry, and try and get some kind of principal component out. Okay. So here's 3 trees. Thanks to Anne and Arndt for a data set. I'm going to talk about 2 data sets today. This is the first one. There's just 3 trees. I think the whole data set is about 120 gene trees. So this is just to illustrate what's going on. You know, looking at that, we've got yeast here next to the insects. Over here, it's in a different position. Over there, it's somewhere else again. And it's not just positions that are varying there. If you look at the primates there, we've got human, chimpanzee, and gorilla. And they're very short branch lengths, but the branch lengths vary a bit. So over there's a much longer branch for gorilla. So there's variety in the shape of your trees and in the branch lengths. Okay, so why do PCA? Well, what can PCA tell you? Can tell you which features are varying most in your data, how features are correlated, and standard principal components analysis also produces a proportion of variance explained by principal component. Okay, so we're hoping our analysis can do all of those things. And in particular, I'm going to be looking at branch lengths as well as shape, because it has certain advantages. Okay, geometry and topology are interdependent. The second data set I'm going to look at is a long branch attraction kind of simulation. Long branch attraction is an effect where branch lengths can affect the topologies you get out. So there's a kind of relationship between branch lengths and topology. Okay. Also, differences in geometry offer a finer resolution of comparison. Okay. So let's just look at a kind of first go at doing some principal components analysis. We've seen what splits are previously. What you might think about doing is taking, for a set of M taxa, we've got big M possible splits, and big M is a very big number. But what we could do is take R to the big M and represent each split with a basis vector, and then what we're going to have is that each tree can be represented by its vector of branch lengths in this very high dimensional space. And so we've turned each tree into a vector, and we could do principal components analysis on those vectors. But the problem is that we've got very few branches in each tree, very few positive numbers. All the rest are going to be 0 in our vectors, compared with the size of the space we're embedding in. So we've got this very sparse data, and the principal components you get out don't really correspond to trees. So what we want to do is do something kind of intrinsic to the geometry tree space. So let's go back and think about what PCA really is. We've got a whole load of points. Those are our trees, and they're sitting somehow in tree space. And, well, standard PCA does something like this. It finds a center for your data, and ideally you minimize the sum of square distances to each of the data points. You then consider lines going through the center, and you project your data onto the line, okay? And then you identify the line which maximizes the variance, or in a Euclidean, sort of in the standard PCA setting, that's equivalent to minimizing the sum of square distances. So we're going to do the same thing as this, but working in tree space, and in particular, we're going to use the geodesic metric. So that hasn't been talked about yet this workshop, so I'll introduce that if you don't know what that is, okay? So that's really what I hope that the kind of picture you're going to take away. I'm going to go through the details of how I do this. Maybe that's, it doesn't matter so much if you don't get that. And I'm going to show you some pictures of results, and hopefully you'll get a kind of idea of what's going on when you see the results. So here's the actual kind of details of the methods. What I need to do is set up this geodesic metric. So I need to explain tree space to you. So tree space is made up of kind of bits that correspond to topologies. So here I'm just looking at 5 taxa, a simplification. We've just got 5 taxa. We forget about the leaf branches and we just look at the internal branches. There's 2 internal branches. I'm looking at a fixed topology, and you can see that the set of trees is defined by this quadrant in R2. So the red branch length is given by the x-axis, and the blue branch length is given by the y-axis. And as you shrink down to 0 length there, you get a multiplication in your tree. Okay, so that's a bit of tree space for one fixed topology. If we look at different topologies, then these kind of bits, these orthents, are going to stick together along their edges. So there I've drawn just 3 of the possible orthents, and they glue together along their edges, right? So those are the 3 points that were on the last slide. And you can move from one orthent, from one piece of tree space into another by doing a sort of nearest neighbor interchange, and that corresponds to shrinking down a branch length and then expanding out an alternative. Okay, so Susan Holmes and other people described tree space in this way back in 2001, and they proved the existence of this geodesic metric. So the way they did that is like this. They consider paths which are straight line segments in each of these orthents, and then you look at a straight line segment going through different sort of topologies, different orthents, and the length of the path is defined to be the sum of the segment pieces. Okay, so then if you take 2 points in tree space, the distance between them is the length of the shortest such path between those 2 points. Okay, so within each kind of fixed piece, each orthent of tree space, we just get the Euclidean metric. So previously, at the last workshop, 2 people presented algorithms for calculating this distance, because it's quite a tricky thing to calculate. And then since then, Megan Owen has developed an order m cubed algorithm for working out the geodesic distance, where m is the number of taxa. Okay, so there's a picture of a couple of geodesics. I've got 3 quadrants of tree space kind of laid out next to each other, and you get kind of 2 things going on. Here we've got 2 points there, whoops, 2 points linked by a kind of straight line, so there's no kinks in that line because it has to be the shortest path. But what can happen is that points can be connected by this kind of kinked path. Okay, so what do I do then? I look for a center point in my data. I consider lines through the center point. So what do I mean by a line? I mean something that's really a geodesic between every 2 points on the line. Okay, so it's going to look like a geodesic, except it extends off to infinity in 2 directions. Okay, geodesic, technically speaking, is just defined between 2 points, whereas this is an infinite line. I then project all of the points in my data set onto the line by finding the points closest on the line to each of the points I'm projecting, and then I search for a line that maximizes the variance, or not equivalently, because we don't have a version of Pythagoras' theorem. So in tree space, that's not equivalent to minimizing this sum of square distance. So there's 2 different objectives that you can try and optimize there. Okay. So I'll go through the details quickly. Finding a midpoint, I basically use a majority consensus tree, because minimizing this quantity is very computationally expensive. Projection, I need to calculate lots of geodesics, but I can do that quite efficiently. The really hard bit is how to search for the optimal line through the data. Okay, if you imagine your midpoint in your data set, that sits in one of these orthents of tree space. And if you imagine a direction vector going through that midpoint, well, we're trying to kind of pick a line with that direction vector that optimizes the projection onto the line. So for each direction vector, we're going to hit a different face potentially of the orthant. And where we hit a face of the orthant, we have to decide which is the other chunk of tree space we're going to go into. So we've got this geometrical problem of kind of which way do we go from our midpoint for our line, but also a topological problem of how we extend. And the way I get around that is really by building up the principal component, this principal line, one split at a time. So I take my midpoint, I find a split that's compatible with it, and I try and find a replacement. So my split is P, I try and find a replacement P prime. So the replacement is when I'm going to travel through to another orthant. And I kind of do that one split at a time. I'll spare the details, but it's analogous to kind of doing one coordinate at a time in a regular Euclidean principal components analysis. And I use kind of different algorithms for doing this search. Okay, so I'll quickly show you a couple of sets of results. So there's two data sets. The data set from Anne on metazoan species, and then I'll show you this long branch attraction simulation. So the first one I've got it's about 118 trees from 20 species. And we're going to look at these principal components. So how do we look at these lines through tree space? Well, if you imagine a point on the line, then that's a tree. And as I move along the line, I'm going to get a kind of animation of how the tree is varying. So if I start off, that's our first tree. And as we go along the principal component, you can see how we change. Okay, so we'll go back up to the top. And so what you should see there is that yeast is kind of moving around from being grouped with the worms to being grouped with sea squirchiona. Okay, so when we look back at those original trees, you could see that yeast was one of the longest branch lengths. So it's not surprising that there's a lot of variability in the data set. So maybe that's why this is dominating this principal component. So instead of working with the original data set, what you can do is rescale your branch lengths so that the average branch length for each particular split is 1. Okay, so we do a kind of rescaling, and there you can see I've rescaled so that the branch lengths on average are 1. And then when I do my analysis, I move through along my principal component, and what you should be seeing there is human and gorilla here swapping places with chimp. Okay, so human and gorilla moving around with chimp, but at the same time you've got kind of rearrangements going on in other bits of the tree there, like the fish there are kind of moving around. So there's some kind of correlation. So the final example then is to do with long branch attraction. What I did was I took a tree with some very long branches on, and I simulated some alignments. So long branch attraction is an artifact whereby long branches in a phylogeny tend to kind of be attracted together, sucked together, and be placed close together in estimated trees. So I generated some synthetic genes just using seqgen from this tree taken from this paper. So you can see there are actually 3 long branches here. We've got a long branch to the archaea outgroup, and then we've got 2 long branches here, microsporidia there. Okay, so when we then do our principal components analysis, I'm showing you the version with the normalized data here. You get something slightly different for the non-normalized. And as we move through, what you should keep your eye on is this microsporidia group. So that's floating around to be grouped with the archaea. So you can see microsporidia is kind of attracted to the archaea in your data set. And there's also a little bit of movement there from the other long branch there. Okay. So just to sum up then, I've had a kind of go at doing some version of principal components by thinking of it as a geometrical procedure in tree space. A crucial feature is I'm using geometrical information as well as topological information to do this. It's a computationally intensive problem to find these lines, and using these examples, hopefully I've shown how we can kind of capture interesting things in data sets. So what I haven't done is tell you about higher components. I've only spoken about the first component, and I've kind of put that down as further work, even though I've got sort of some results along that line. But I think what I'd like to do is really get a better understanding of the theory of distributions on tree space, as to whether really thinking about distributions as kind of rugby ball kind of spreads of points is the right thing to do. So finally, I'd like to thank Anne and Arndt for the data, and also two anonymous reviewers who were very helpful in kind of shaping this method when I sent it to review. So thanks to them. Okay.
I didn't catch why you would normalize the data. If what you really want to see is what's happening on the tree, why would you want to get rid of the branch length data?
The reason for doing that is because, for example, suppose you're interested in variability in the gorilla branch length. You could see on the original trees that gorilla was basically much shorter than yeast, for example.
Okay.
And so it can be changing a little bit, but you're interested in that variability, and so you kind of factor out by the average of its branch length. Yeah.
In standard Euclidean space, you have the Malhelin-Obis distance that's used when you've got scatter that has different variances and different dimensions. Is that sort of the kind of normalization you're trying to achieve?
The normalization I think I'm doing here is a bit more like analyzing a correlation matrix instead of a covariance matrix. That's the kind of idea, yeah.
So when you do this analysis, you walk along the principal component, those intermediates weren't any trees in the data set, and nor were the endpoints. So is there some way you can convert this into a sort of support vector idea where the actual trees that you show are real trees from the data set?
Well, what you can do is you can project each tree onto your principal component and produce a table of distance from the midpoint and distance from the line. And so you get a kind of decomposition of your data in that respect. Yeah.
No more questions? I mean, obviously it's going to get harder and harder when you get up to your next stage, looking at two or more. I imagine it's not just going to be more difficult computationally, but just forming an idea. I mean, so is there any mileage in just sort of going back to the Euclidean space and then saying, okay, let's do a principal component analysis in this? Admittedly, the real trees only live in a tiny part of all of this, but would you then be able to sort of project out of the illegal parts and then into a sort of legal part? Would there be any sense in that kind of approach?
I don't know. Yeah, I mean, it hurts my head a bit to think about planes, but the kind of thing I've had a go at is building two principal components sort of at the same time. But the problem I have with approximation by planes, I'm not really convinced that if a sort of best plane exists, it might not contain the best line. So, yeah, it might, but it's not clear to me that it does. So, yeah.
All right, thank you again.